Okay, good morning. So this is a week 11 computer session. So today, so let's start by updating the toolbox again by reinstall it. Okay, you know where to go and copy this code and come back to the same folder and then just overwrite the current folder and there's some behavior change for some of the steps. So you want to have updated version. So we're on the same page. Otherwise, what I demonstrated here, we made different from the older version you use on your computer. Yeah, so I update every week. So if we have some problem, okay, we keep updating it. Yeah. So follow what we discussed during the lecture. So today we are going to look at the CCA analysis, then DTW, dynamic time warping, and then I will lead you to revisit highly variable gene detection methods. So today we are going to look at two different methods. One is the one used in serial package method. Another is the method specifically designed in single cell splitting fate. These two methods may produce different results, and it's common. I will share with you some of this paper. Other people evaluate the different methods, generate different method results. And then I will introduce another gene function annotation tool. It's called metascape. Okay, so normally we use enricher to do the gene ontology or pathway analysis, gene set function enrichment. Test. But this metascape found is another alternative method which is really good in general. So will be very useful when you do this annotation, whether you found a differentially expressed gene or highly variable expressed gene, what you have to do with it. And then we will come back to look at a program called Harmony. So this is for batch effects or remove control, okay? Oval. If you have cells you believe they should be in the same group, and you can use this. We introduced neutral nearly neighbor and also CCA method. This is another one which is very popular. I didn't mention it during the lecture, but this method is available in the package. So I will we will see how we can run it. The problem with this is Python based. So we need to have our Python environment installed. We did this a couple of weeks ago. So suppose you have that. At the end, we will look at the monocle. Monocle, we are talking about monocle2. Recently, there is a bug, and a lot of people just cannot run it's online talk about this bug. But let's see if we can successfully infer some trajectory. But my guess is we don't have such a luck, and we may not be able to run it. Okay, so let's come back to the documentation of canonical. Okay, so this is a CCA analysis, canonical correlation analysis. So we will just use the example data provided by this documentation. So the idea is you have two inputs, okay, two matrices. So they are from a different sample, measured in different way, but you believe there's some internal, unspoken or latent alignment. So you want to find this alignment. You try to rotate X and rotate Y to let X become A, okay? The input of X is a matrix. Output A is corresponding matrix after the rotation. And Y is another input matrix. And its output matrix is B. So we rotate both A and Y, X and Y respectively to allow the output A and B are aligned matrices. Their correlation are given in this R, so it's the correlation coefficient. If you ask more, there are U and V. So U is a rotation matrix to rotate X. V is the rotation matrix to rotate Y. So if you use U multiplied by X, you get A. If you use V multiplied by Y, you get B. Okay? So if you ask other intermediate steps, there are other statistics you want to keep, you can still get those. So we are going to just use this example. This is from a built-in data set. It's a car, bigger car. And then let's construct a data. I just copy this and put the whole data matrix in a variable called data. So this example will create X and Y, but in this example, X and Y was measured for the same sample, but it doesn't have to be. Okay, next step. This data set contains missing values. So this step is necessary. We just remove some samples contains missing values. Now, if you look at this current sample, 406. If you look at them, and the sum of these rows contains non-NaN, right? This is missing values. So these three lines detect which row you have at least one NaN, then you found those row index and remove them. Only keep these rows that do not have NaN. And also for variable 1 to 3, Okay, the first 3 columns have been assigned to X, and the 4 and 5, the last 2 columns, are assigned into the Y. So we have X and the Y. Now, if you look at the dimension of X dimensionality, this is only 392 samples. It was over 400, but some of them contains missing values. We don't need them. We cannot use them. And the Y is the same sample, but defined in 2 other variables. So look at their what each column means. So this is displacement, the horsepower, and weight. That's in the first 3 columns in X matrix. Acceleration and MPG in the second matrix, Y. Yeah, so we just call this function by asking for I think I made a mistake. I thought A and B are the rotated data points, but no, A and B are the rotation matrix, where U and V are the outcome of after rotation. So CCA by default, if you only ask for first 2 outputs, they are A and B. So A is a 3x3 rotation matrix. The reason is X is defined by 3 variables, so it's a 3D data set. So you rotate this 3D data using 3x3 rotation matrix A. So Y is a 2D data set. So B is a 2x2 rotation matrix. Okay. So the U is the after rotation, what you get. So if you look at the U, you have a 392 times 2. Okay. V is, so it's reduced to the 2-dimensional for both matrices. And R is correlation, okay, for each variable after rotation. We can jump to, we don't have to look at them. We already show the whole matrix A. You can copy this part and get this figure with the 4 panels. Let me show you how can you make those panels. So you plot, you define this hide layout, and then tell them this will be 2x2. So you have a figure with two rows, 2 columns. And then you say, okay, next tile, and it will automatically put your figure. So you have 1, 2, 3, 4 figures to plot different values. And this output for first dimension of the output, the second. Okay. Okay, so UV is the first variable after rotation. So the correlation looks like this. Okay, you can figure out, well, this U2 and V2 for the second variable after the rotation, positively correlated. Well, for the U1 against V2, there's no correlation. They're orthogonal somehow. Okay. Similarly, for U2 and V1, there's no correlation. So this is just give you some idea about this outcome of this CCA. You can imagine if you plot those points after rotation on the 2D space, they should align because they both U1, V1, U2, V2 are how to say they're correlated. So I'm going to plot out these points using 2D. Okay, let's see. Close all. So if I do scatter, each sample we have U, U1, U2. Okay. So let's just type this. So U is the rotation for the X. Okay. So I put a hold on so I can come back to plot V onto it. Okay, so that's my scatter plot, but the two scatter plots superimpose onto each other. So we should have this so-called aligned outcome. Remember, those blue points are from U. So there's one matrix to define 392 cars. And then V define same cars, but use different variable. But they are not aligned in this. If you do X and Y to replace this X, X2, Y, Y2, okay? As a before alignment. And also we lost some information because X was 3D, but we only used top two. But that's unaligned data points. Okay, so you see the effects. So after you aligned it, you have somehow also normalized it, right? So those batch effects control programs just use this CCA to try to say, okay, if you have a two-cell batches supposed to be aligned, you should try to force them to rotate and align to the same space. Okay, any questions? I found this technical paper really, really good to discuss a lot of those, including the CCA background. Let's find those part. So the problem we are talking about for the CCA actually is something like this. So you have a remember A and B, and those are the rotation matrix. Yeah, this is very okay. I will talk about this later, maybe. Okay, so let's stop this, close all, and we are going to look at the another function, DTW. So this function, let's see, which, no. So for example, the CCA function is inside the statistic toolbox. This one's also in there, DTW. So it's, oh, this one is in the signaling processing toolbox. We are going to go through small examples they provided. Okay, so this example is simple, and you just copy this and then do DTW and give these two signals, let them to align. X and Y. Okay, this is can be used to compare like two curves. If you have two curves, so you see, okay, what's the difference between these two curves? If you compute the distance between each points on this blue curve and the red curve, certainly there's a large difference. Euclidean distance should be large, but if you estimate after a line those signals, the distance is much smaller. Okay, so especially if you have two curves, you have no idea whether they are alignable or not. Maybe you try this, and the similarity between two signals may be recovered after you do this time warping alignment. Let's see other examples. Okay, if you change the curve, the second one, then you get a different distance. Okay, so the alignment of these two wasn't as good as the first pair, and this not nicely aligned, so the distance become larger. You can also try the last example with a 3D because they're imaginary portion in the signaling. I think you can just use these small examples to get some idea, and maybe sometime later you have the problem, you know, okay, there's a way you can align this theoretical or some kind of structure in there. You should be able to stretch the signal to make them aligned rather than just directly compare this to okay. Any questions? Now I'm going to show you two papers, highly variable gene related. So the first paper is this evaluation of different tools for highly variable gene discovery. Okay. Let's see. The other paper is also evaluation paper. That's to compare gene features, selecting gene features for unsupervised analysis of single-cell gene expression data. Okay. So when they talk about the featured genes or select gene features, what that means is to find highly variable genes from your given single-cell RNA-seq matrix. And to select those, they called feature variable or feature genes to help you to reduce the dimension, because otherwise you have 10,000 genes, is too much. You may just select top 2,000 highly variable genes for the downstream analysis, for clustering, for cell type detection, etc., etc. So the first paper, the key idea is there may be 7 or 8 different highly variable gene selection algorithms, and the outcome, the gene they selected for the same data set is very different. Okay, there's no very little consensus. So what they use is use the package called basics, use Branek, this paper published in 2015, and use another paper, Surrey package. I don't know which one they use. Okay, so they compared basically 7 or 8 different algorithms. First, the number of genes detected is very different across all the methods. Okay, we have different data sets, but some say, okay, there are more than 20,000 genes. I can hardly believe. But anyway, so you see this difference. There are very little overlap to the major category of methods. So you see this Venn diagram. Okay. So in general, what we can tell in the field, there's no consensus say which gene supposed to be real highly variable gene. So it all depends on your method, the way you select your cutoffs, what factor you consider. Okay, now to come back to this paper. So this paper is 2 years later. So that was 2019, this is 2021. So this is the method they evaluated. You see some of those we already seen from the older paper, like a basics here, Branek, okay, HVG. So they also include this the MATLAB toolbox, the method we developed. Okay, so they include more methods, and all some belongs to different categories. So I want you to spend 1 minute to read their description of the method we developed in the toolbox. So what we consider is a 3D. We consider both average gene expression for the gene across all the cells. We also consider its CV, okay, coefficient of variance, basically is a dispersion. We also consider the dropout rate. Okay, see, this gene expressed in 95% of cells, another gene maybe only expressed in 15% of cells. So we use these three combinations of their gene expression mean and the CV and its dropout rate to define each gene in this 3D space. Then we did a regression, some kind of regression to find majority gene will follow a curve. And then this curve, all the points is supposed to be that gene's expression, the expected value. And then we found, see which gene is far away from the curve. Those distance was used to as a cutoff to select highly variable genes. So our method is very different from all others. Then we start to do the evaluation, say, okay, for the same data set, if I use Brenneck method, I use toolbox method, they select a different set of the highly variable gene. Then for the downstream analysis, which set they selected better. It's difficult to see, but they use some criteria to see, okay, this method detected more informative highly variable genes for downstream analysis. I don't know exactly how they did that, but what they found is a different data set A, B, C. ARI. Yeah, so sometimes for a lot of genes, so you see this toolbox really behave pretty good in the first two data set. They consider to be the best. For smaller data set, it's not. So anyway, I believe our method is some kind of competitive ones across all this. Okay, I'm not quite agree with how they use highly variable genes. So they just use highly variable gene as a step to reduce the number of genes to include it for the downstream analysis. They did clustering, the other things. But we use highly variable gene to understand homogeneous population of cell to understand the function. So we are not dealing with clustering anymore. So, but it's a different purpose. But let's see how this can be done in our toolbox. So in the toolbox, we implemented two algorithms. The one is I just mentioned, use a 3D to find highly variable gene. Another is Brenneck, the method that I described. Yeah, I think, yeah, it's this paper, okay? The method is use the first author's name, surname as a name of that method. Okay. Now let's come back to the MATLAB and let's see how we can access these two methods to detect highly variable genes. Let's just do a clear all. Okay. For a quick demonstration, I just use this small example data. But in your real application, you may find more genes, more cells in the same group. Remember, if you want to study the function, you have to keep the cell number enough and also homogeneous. Okay. Let's see today which group we are going to use. Let's try this number. So I just want to find which group I want to do the demonstration. Well, this small data set is really not ideal to do this. Only have 2,000 cells, and also in different clusters. I know the alpha cell has the most cell numbers. So what I can do is just extract our alpha cell with cell type. Click this button here, select cell by class. And select cell type, and then I know alpha cell is the most abundant. Select. Okay, so alpha cell in this example has been clustered into two separate groups. So they may belong to different subtypes of the alpha cell. Ideally, we should select just one cluster, okay, to do the highly variable gene detection for function analysis. But in this case, because the number of cells too small, so I just keep everything here, okay, just treat them as a uniformly expressed group. We need to filter out lowly expressed gene by, say, click this button and remove gene by expression and select 0.05. So gene has to be expressed in at least 5% of cells. Then if I click OK, I remove 384 genes. Are you okay? So the steps, any questions? Okay, I just make this, remove some extremely lowly expressed gene, okay? I keep same name sample cells. Otherwise, we don't have enough cells. So the function is this. Remember, there are several warning messages. We have been going through this. The function of this button is to find the highly variable genes for your cell, and then to do the functional enrichment analysis. And then another warning message is the function applied to a homogeneous population of cells. For our purpose, we're not to extract feature genes for clustering or others. So we know they're same cell type, at least that's okay. We already removed the lowly expressed gene, so we can let it go. So at this stage, you have two options. We are going to try redneck this old version, old method first, then you can try this sublime fit method, which only available in this toolbox here. Okay, you see this, and the method will find the genes which way above this curve, okay? Those are the residual CV to indicate how variable, the level of variability controlled for the mean. So you have to check whether this curve fitting is okay. So in this case, not ideal, but largely okay. We found a lot of, okay. So we just save. So if you have more cells, this estimation may be better. The regression will be better. So now we just save this output to a table in the variable t. Then we are going to select the top, say 200 highly variable gene to send to enricher for the function analysis. Okay, the top 200 highly variable gene to do enricher search. So on this page, the selected top 200 genes has been pasted here. So I'm going to make a copy. I select this to make a copy because I'm going to not only use enricher, but another, the metascape, to do the analysis at the same time. So after you copy this, you can submit and to get enricher results, okay? Especially I will look at ontology, pathways. Remember, you already copied the 200 cells, the gene names. You can come to another tab. In this case, we are do metascape analysis. Okay. So we are doing two gene enrichment analysis using two different online tools side by side, and see whether they have the same similar results. Okay, what I can do is here, let's see, I want to paste a list of genes. Let's submit. This is from a mouse. Okay. So it also gave you a number 200 because they match all the name of genes to the mouse gene name, and 200, all the genes has been matched. If you select the human, there are 194. So there are 6 genes which are the mouse gene, but not in the human. But this one is 200 genes all available. So then you click analysis to let it go. Are you okay? That's fine. What we did, we just select the alpha cell from the sample, the small sample, found the highly variable genes, and gave this gene list to 2 different websites. One is enricher, another is metascape. Enricher is really really quick. You immediately get this outcome of different pathway enrichment, gene ontology enrichment. For example, pathway, let's see, in the rectome, then you have the top signal transduction pathway is this, and the gene in our 200 genes and also in this pathway are highlighted in this black box. Maybe we should look at what this pathway is. You can visit this web page. Search for this ID. So I have no idea what it is. So this ID is a process with extracellular signaling, ELISA change in cellular states and transmembrane membrane. Okay, so that's what our alpha cell is doing with a lot of genes in this signaling pathway become highly variable gene. I mean, they must be very, very active in terms of this. And the second one, interleukin. So this is the cytokine signaling. So this too is consistent with what we saw in the gene ontology. Okay, in gene ontology, you see the top ones are the response to cytokine stimulus. Okay. So this actually is a little bit strange because we don't know why alpha cell have this activity to respond to the cytokine stimulus. But later you will realize those alpha cell was collected from a type 1 diabetes mouse model. They're not a healthy mouse. They're by nature, they're TID, TT1D, type 1 diabetes mouse. Let's come back to metascape, see what we get. Okay, so another report page. So it's finished. So we can click the button to see the report. So for the same 200 genes, and we gave to metascape, it gave us this summary of which gene ontology or other gene set the most significant. Okay, those are negative log 10 p-values. It's weird. I have no idea what they are doing here. Okay, this summary and also I think this is only top 20, and if you go top 100, you get this. If you only need ontology, so remove other data sets, you have a picture like this, only contain the gene ontology terms. Let's move down. So you have your input gene list here, 200 genes. Okay, basically those are the tables you generated and to support the figure, the bar plot you generate, you see in the summary. So there's the tables, and here you start to see this enriched term network. So they build a network for enrich the term. If they have a term very similar, they group together. For instance, those blue terms may be all related to the regulation of the wound healing. You can visit this interactive cytoscape website and have a network like this, and you highlight each spot here, you will see the name, okay? The name of the term. And they have a PDF version of this figure, so it's ready to be published, right? If you found our results is really interesting. And then they also provide this protein-protein interaction enrichment analysis results from strain database. So they map the gene into their interaction networks. And within this, it's found small modules. For example, this green module includes 3 genes. And this larger, and this one contains 5 genes. Okay, they're supposed to be a protein complex, giving the information they found from a protein-protein interaction network database. And they also have this table. This is a transcription factor database. They probably found the set of 200 genes, a lot of them under the regulation of the TRP53. So that's supposed to be a top transcription factor, give you the activity of the change in the 200 genes you selected. At least so there's a significant top key regulators. And here is the detailed information. So in 200 genes, you provided there 15 are regulated by TRP53. 16 are regulated by SP1. So, okay. Then they compute a p-values for those transcription factors, so as a key regulator, how significant they are. Not only by the count, okay? So this 15 have a more significant value than this gene control 16 genes. The reason is the TRP53 targets maybe smaller, the number of smaller may be smaller than this SP1. SP1 may be a more general, universal regulator, regulate a lot of genes. So it's not so surprised if you see 16 gene and its control. Anyway, so that's the metascape provided. So it's slightly different from what you get from enricher, okay? Yeah. Why want to do this? No, whenever you have a set of genes, say you have 200 genes, you do whatever analysis you get this gene out of your 20,000 genes. So you want to know whether these 200 genes are functionally linked, okay, enriched in certain part of this cell function or molecular function. If these 200 genes are randomly selected, there are no association. Then you get very noise information from this enrichment analysis. In contrast, if they all move towards several specific function, you will see, okay, those function jump out. And then you know, okay, the set of 200 genes are not random sets. They are enriched for some of the function. In our case, we're using highly variable gene to see what the alpha cell is doing in their active. Okay. So we will take 5-minute break and come back to use another method to detect highly variable gene. And I will compare, say which, number one, I will see whether 200 gene detected by this method and that method, how much overlap between them, okay? And then we do the functional analysis again and see whether there are differences. For the students, I'm going to give you this as the last homework, which allow you to select a cell type you think is worth trying to analyze their highly variable gene and understand the function of those genes and to infer what the cell type in that situation is doing. So you have to go out to find the interesting cell type you try to work on. So you may go geo database to search keywords, to download and to get the homogeneous population of those cells, and then to run 2 different highly variable gene detection methods I use, and use enricher or metascape to understand the highly variable gene sets. So now I'm going to show you the second method. So we still work on the same population of cell, and then just assume we already treat everything. And although there's another highly variable gene, highly homogeneous population, but it just as a small example, we just keep doing this. Okay. So I'm click this and just go through those warning message, and then select the spleen feed method. Okay. Okay, so now we have a 3D, and you see the mean is log average expression of each gene across the cells, and then CV is their coefficient of variance, and then z-axis is the dropout, how many zeros basically. So on the top here are the genes. For example, this one, the drop-off rate is really high, okay? It's close to 95. So basically, this gene is lowly expressed, maybe only present in 5% of cells. So that's why drop-off rate is so high. And correspondingly, its average should also be really low. So it should be in this end of the mean. And the CV should be higher, okay? Because they're so sparse, and then CV is really high. Once you have that, you have a really high level. I don't know, on the screen you can see this is each point is a gene, okay? I made this feed it out so you can see the underlying curve go through the majority of genes to evaluate how good this fitness is, the fit of the curve. If the case you have a very bizarre case, the shape of this curve, that may indicate you have not processed your sample, your cell population, or remove gene sufficient enough. Otherwise, you should get a very smooth curve. So you can evaluate this curve fitting, and if you think is okay, then we start to use this curve as an expectation. So you expect if you have that level of dropout, you should have that level of mean, that level of the CV. So you fit majority points on this curve. But now look at those genes, which, for example, like this, okay? This jump out far away from the curve. That means this gene have some unique feature in terms of these 3 parameters. Either it have a higher than expected CV or higher than expected dropout rate, whatever, okay? Whatever reason, this gene is far away from the regression curve. It's not a regression, it's a spleen fit, but same idea, okay? We track the majority of the change across most of genes. If you are outlier, okay, like this, and there are some of them genes. I don't know if you can see there are some points here, okay? So those genes, I will call them highly variable genes. For whatever reason, you either have a more than expected dropout, more than expected CV. So this method is data driven, okay? It only depends on majority of genes, what the shape it is. So your sample, this sample may be different shape of the curve from that sample, but it doesn't matter. You only consider all the genes you measured in this particular sample. So how can we select top highly variable genes if you have this curve? After that, you should select this button to highlight, say, top 200, and this will find those genes, okay, based on the distance to the curve. Okay, so once you have that set of genes, you can send the gene list to the enricher analysis by clicking that button. The 200 genes will be carried to the web page of the enricher, okay? Then the rest of them should be the same, okay? You did for another set. Remember, we saved the 200 genes from another analysis into a table T. So here we can do the same to save these 200 genes, to export the names to T2, okay? I don't know if you have that, but I have this, oh, this 200, yeah. So the table is here. It's sorted by, so this is from method 1, okay? So method 1, I have a table, a gene, this 200, from 1 to 200, this top 200. So this is my gene list 1. So using the first method. Then my G2 is this T2 is here, okay? But anyway, you have a way to get the name of gene out. We want to look at these two methods, whether they're top 200 genes, how much the overlap is. So once you prepare this to list of strings, you see this is the G1, this is G2 list. We can do intersection, intersect G1, G2, gave it to G12, okay? Look at the length of G12. So G12 contains 116 genes. So it's really good, actually. They are shared 116 genes by two methods. Okay, 200 here, 200 here, 116 overlap. Okay, so that's the method you use to get some idea to what are the intersect. And then come back to this page. I'm going to submit this to enricher and see what ontology we get. Yeah, I forgot to copy the gene name, so I should copy this 200 gene name. And because I'm going to send it to Metascope, I'm not so sure this program interface is smart enough to get rid of those. Okay, it's good. It's called quotations. So 200 genes have been found, and then express analysis. So I moved really fast. You may have some trouble to see, okay, how can you get these 200 genes from this interface? But eventually you should be able to figure out whatever the way there. The gene is here, so you can see or either a copy or something like that. But that's a function you want to use to, once you have the name of genes, to find overlaps. I think for enrichment analysis, 100, 150, 200 is all fine. You can try and see, okay, if I use top 100, whether I got highly similar results from 200 genes. There's no fixed cutoff. I don't know. Yeah. Yeah. Yeah, you can try both methods. Yes. So let's see the report generated by Metascape. So the top one, enrichment gene ontology response to type 2 interferon. Okay. I think the overall pattern is the same, but this is the most specific to me. Okay, it looks straightforward to the cytokine signaling infections. Yeah, top this transcription factor are the same. TRP53. Remember, we have the same, and there's two others. Okay, the SP1 in this first position, and there are two others have been identified by this new set of highly variable gene detected by this 3D method. Okay, so we don't know whether this make any sense or not. Okay, come back to our genes cells. So those cells are alpha cells. We don't expect they have this a lot of immune cells function. So why is that? So if you look at the metadata, and these cells are collected from these two studies, the same study of two samples. So I copy this, and it's in the metadata information. Okay, I copy this and search where I get those samples. So it come back to me. This is pancreatic cell. Okay, pancreatic islet cell. So the alpha cells there, the strain they used is this. If you search this mouse strain, you know this is a type 1 diabetes model. So their alpha cell in the pancreatic It's not in the healthy state. It's probably under attack with some T cell around it. So that's why we found those cytokine response signaling. Okay. So if you, my hypothesis is if you don't use this type 1 diabetes mouse, but use healthy mouse alpha cell, you don't get those signals. If that's the case, it's really strong indication that highly variable gene to select not the cell function, but also the active cell activity under its current environment. Okay. So your homework probably will be go to find the alpha cells in the healthy mouse and repeat the same analysis. Okay, see, no immune response gene active in there, then it's a good story. But you can select any other cell types you want to work on this. Okay. That will be homework number 4. I think that will be the last homework in our semester. Any questions? Now we don't have a method to compare two different conditions. For example, this sample is from different weeks. Okay, this is 8 weeks. Another sample is 14 weeks. Another sample early. So can we do this highly variable gene detection at different time points and see whether, okay, some genes always highly variable expressed across time, but some new highly variable gene appeared in the later stage. How can we do this analysis? I have no idea right now. Okay, we are trying to do this to compare cross sample and to detect differentially highly variable expressed gene. Okay. Right now, what we can do is just do this separately. We detect highly variable gene in week 8, highly variable gene in week 14, and see what's the difference between them. But they're supposed to have a better way to do this. Mix the cell. I don't know if it's a good idea. Yeah. So when you mix the cell internally have 2 subclusters, and they are not homogeneous population anymore. So this highly variable gene method is supposed to apply to homogeneous population. So it's a good question, but yeah. So let's see. All right. So that's a homework and also highly variable gene, 2 different methods. Okay, you can play with this. Now we are going to move to harmony analysis. There are a lot of packages called Harmony. If you search Google Harmony Python, you get different packages for different purpose. So the particular one I'm going to use is mentioned. It's about in this paper. Okay, the algorithm developed it. So this is an overview of this algorithm. You have several different clusters by its own, but also within the cluster, there are different batches of data. So green, purple, yellow, they're supposed to be aligned to each other within the cluster. So you start to do this iteration until, okay, they move together, but they still keep the original global structure of the clusters, but it just put different batches into together. So our input for this algorithm is their embedding, okay, their position in the 3D and their labels. So I know which cell is from batch 1, which cell from batch 2, which cell batch 3. You probably can give the cell type, okay? So there's no big X involved in this algorithm. So no gene expression level or gene name play no roles in here. So this is purely data driving game, okay? This is a data science game. The only input information is the position of those cells, okay? Coordinates in 3D. And then their labels, the categorical labels, that's a very, which cell belongs to batch 1, which batch 2, batch 3. Okay, so the package I'm going to use is implemented in Python. So, you know, we have already installed minicodon here. So we need to bring this up in order to install the package before we call it from MATLAB to call that. If you don't have this, you have to review previous lectures. I should have recording. We installed this, and then you have this interface. You have to pipe install harmonypy. Okay, this is a package. The minicodon basically is environment I installed here locally, and then including the Python environment inside there. Are you okay with this step? Okay, I think we are done. Now we come back to our example data. Okay, this is from a mouse pancreatic at different time points. So in here, we only have, if you look at the batch ID, you select this to highlight batch ID. We have 2 batches. One is 8 weeks, another 14 weeks. I should include more. And remember the data set, we have another week time point. Okay, anyway, so I'm going to give this information, so the label of cells in 2 different batches to Harmony, that Harmony to move the cells if they belong to same category, same subcluster here. And this, for example, those cells should be moved to those cells close together, okay? Because they may belong to the same cell type, but now they separated. Are you ready, everybody? No? Not ready. Yeah. Anyway, so once you finish the installation, that side you should be able to come back to this external function and select the batch integration using Python. I just quickly demonstrate and see whether you can get the same outcome. So I click this and allow the algorithm to run. So remember the position of those cells. It will, after that, it will start to move. Okay, now they move close to each other now. Then we can run that again to the iteration. So I'm not ready to save this embedding. I'm going to rerun this. I say no. Look at the position. They move close to each other a little bit now. Let's move here, okay? And then I run harmony again. You can repeat this process several times. You will see this start to merge a little bit. So okay, compare your original position. Let's see if you can get it. Okay, that's good. Amazing. Working? Maybe not. Yes. Oh, you haven't installed it. Let's see which one you are using when you code on Python.
I think we had two. I had two versions.
You installed two, yeah. You point to the wrong Python, yeah. I think you can select if you know where it is. Yeah, so select. Use another, then you select. Yeah.
Data set is not exactly that's the one.
But it's working, right?
Yeah,
Yeah.
but the data is not exactly that's the one. Yeah.
It's different from what I get.
Yeah.
But you look, you get a better results. You have a complete. You just run once and you get this. Okay, good. It's good. I don't know. Yeah.
So like when you repeat your run, do you hide
You send the current position again to rewrite it, make it even closer. Yeah. Yeah.
This is from the beginning.
Oh, you use a different embedding. Yes. Yeah. So you start from a T-SNE. We start from slightly different. We start from if you click this, you better. Oh, you use old version, so you haven't updated.
Oh, okay.
Yeah, yeah. You use older version of the toolbox. Yeah. But it's the same idea, okay?
Yeah.
Same idea. So this tool may be useful when you really require this remove of the technical batch. And you know you should remove them. That's okay. So make the picture looks better and your PI or may happy if you have no batch effects across different samples. But as I said, there are no change in the big X. So the expression level, no change. The downstream analysis based on the expression level matrix will not impact it. You only change maybe the clustering results. So if you have this, when cell moved together closer, then they may cluster in the same group. Okay, they may you may improve the cell type identification because your cells in the same group clusters change the composition by after move of the harmony. So it's somehow useful, but I don't think it's deserve a nature biotechnology application, nature methods. Yeah, so the last program you can try will be pseudo-time analysis. You have this and you just click this. And check the error message and ask the authors until they fix. Okay. So the first time you run it, the program will be automatically installed from the back end because the MATLAB detect monocle2 has not been installed. So you see the start to install all the dependence and monocle2. Yeah, so I ran into the problems. It was working before, okay? So you check the error message, Google, and what the problem is. So the error in so if you Google this part, you will find yeah, so this one is a recent problem, and I think the solution is somewhere here. But anyway, we have to wait until they update their package. Okay. So I cannot demonstrate the application of this today until they fix this. So it's very recent. arrows. What I did again is I copied the error message here. Okay, copy this name of this function, and then I search Google and found this issue. And in the monocle release there, I hope they can fix this issue very soon. I think I will use last 5 minutes to demonstrate another function here. We will use this function for next week's lecture. So this is called a KNN graph. So you have a cell here, and then we are going to look at each cell's neighbor and to make a link. So here is a cell KNN network. If you use 3, you create a KNN network like this, the shape. Okay. Is this 3D or 2D? Yeah, this is supposed to be 3D, okay? So look at this region cells, this part here. They form a triangle here because you use a K equals 3, so they try to find each other, the nearest neighbor. Let's use K larger than 5. Okay, you get a slightly different picture. There's no analysis will be done with this, just a visualization tool, okay? To give you some idea about it. So if you have a very complex structure, you can use these small steps to go through a very irregular shape of your cell cloud or other type of this structure, okay? All right, that's all for today. We want to share any other questions. I will set up this homework in the Google Classroom with instructions, with the 2 papers I can copy to there. Then it's up to you, say which cell type, which data set you want to use to check, find some interesting topics to search, to do research on, okay? Okay, I'm going to stop recording.